Well, good morning, good afternoon, good evening, everyone, and welcome to the world's most exciting classroom. My name is Joe Grabowski, and I'll be your host for today. This will be our last event together for a couple weeks for two reasons. One, I'm going to head to South Africa on Friday and catch up with the Oosterschelde and the DARWIN200 team, and we'll be doing some live events from the field. But the second reason is that a lot of classrooms in Australia and New Zealand will be on break for a few weeks after this week. So we've got one more hangout together in the world's most exciting classroom before we take a little break. And then in a few weeks, we'll continue where we left off. And the Oosterschelde will likely be in a different location, maybe St. Helena, if that's when we get things going again. Really quickly, I'm going to share my screen and I will show you a quick update on where the ship is. Spoiler alert, it's in the exact same place where we left it last week. So the ship has arrived in Cape Town. It is going to be here for about 3 weeks. We'll have our young Darwin leaders in the field. We're going to have different events going on in Cape Town and Hans Bay. I'm going to be in Kruger, Greater Kruger area. So we're going to have a lot of stuff going on. Then in about 3 weeks from now, the ship will set sail and make its way all the way up along the coast of Africa. A few stops along the way in St. Helena and Ascension, the Azores, and then finally in July, we'll return back to Falmouth after 2 years of traveling around the world. So that's a quick update. That's right where we left the Oosterschelde last time, and we will catch up with Josh. We have a great recording with Josh in the harbor, and we'll play that after our first guests join us. So our first guests tonight are joining us. Oops, I should come back from that map. There we go. Are joining us from New Zealand. It is so much fun when we get to visit the North Island of New Zealand and spend a little time with Emma at the National Kiwi Hatchery. They do incredible work there. As some of you may know, the Kiwis are flightless birds. They're endangered, but thanks to the work of Emma and her team at the hatchery, they are doing their best to bring them back and get them back into the wild. I'm going to bring Emma Bean in from backstage right now. Here we go. Hey, Emma, how are you?
Kia Joe, yeah, really well, thank you, and welcome to our new location. So we are the same National Kiwi Hatchery, but since we last saw you, we've moved to a new site.
All right, very cool. Am I still right? Is it still on the North Island, or did you jump all the way to the south?
No, it's still on the North Island, and we're just 5 minutes from our old location, but we're doing so well, saving lots of kiwi, that we needed more space. So we've moved to a new location where we've got room to have more kiwi growing up to that stoke-proof weight.
All right, very cool. Well, I am going to let you take over in just a moment, Emma, but I do want to give a shout out to the classrooms who are joining us at Norcourt Primary School in South Australia. Just send me a message in the chat. Let me know if you got in okay. I know you're joining a little bit different today, but send a message in the chat. Let me know if you got in okay. And Emma, I'm going to let you take over for a bit.
Wonderful, thanks Joe. Well, kia and welcome everyone here to the National Kiwi Hatchery based in Rotorua, New Zealand. Now, as you might be aware from the learnings through DARWIN200 already, back before humans arrived to New Zealand, there were no land mammals, so it was all birds. New Zealand was essentially birdland with some reptiles, amphibians, insects thrown in for good measure. There were 2 land mammals and they were both bats. So strictly speaking, 2 land mammals, but mainly birdland. And because of that, all of our bird species in New Zealand have evolved in a really unique way. So it means that some of the roles that mammals would typically fill in countries around the world have been filled by birds, and kiwi are no different to that. So kiwi are nocturnal. They are flightless, as Joe said, and they spend a lot of time on the ground foraging for insects and fallen berries. So a similar role from, say, the UK, where I originate from, would be the badger. And so the kiwi is filling those unique spaces within New Zealand's forests. Now, because New Zealand was birdland, a lot of their responses to threats were very different. So natural threats would have been other avian threats like the now extinct Haast eagle. The best thing to do is to freeze. Now, unfortunately, with us humans arriving, the original settlers, the Maori, they brought over the dog and unintentionally the rat, but us Europeans, unfortunately, did far more damage by bringing over the mustelid family. So that's the ferret, the stoat, and the weasel. And we also brought over other things like the possum for the fur industry. And so when people originally arrived and settled in New Zealand, so similar time when Darwin was travelling in 1835, they estimate there were about 12 million Kiwi. Now we've got an estimated population of 68,000. But it's not all sad news, because we've come up with the solution to this problem. So we've got an intensive predator control network around New Zealand's forestry, but we also do something called Operation Nest Egg. And that's what we do here at the National Kiwi Hatchery. We work with projects all around the North Island. They monitor the birds in the wild. They bring us the eggs. We incubate them, hatch them, and raise them to what is deemed a stoat-proof weight of 1 kilogram. And at that weight, instead of just having the somber statistic of only having a 5% survival rate in the wild, by going through Operation Nest Egg, we turn that statistic around, and instead of 5%, it's 65% survival. So we basically keep them safe when they're most vulnerable, release them back out into the forest when they've reached that stoat-proof weight of 1 kilo, and by doing that, we've helped increase the population. And we've actually, in the time that I've been at the hatchery, we've celebrated moving up a threat status. So Kiwi are no longer on the endangered list, they're actually conservation dependent, which is fantastic. It means what we're doing is working, we just need to do more of it. So come with me through to the hatchery and I'll show you a little bit around the space that we do this work in. We do host visitors on Manahiri around the hatchery, and that's a really great way that you can help us support Kiwi. So come and visit and see the work that we do. This is the egg arrivals room. So this is where the field workers bring us the eggs in the first place. Now the Kiwi is about the same size as a chicken, and as Jo mentioned, they're flightless. So they're actually quite a lot heavier than flighted birds. They're part of that ratite family, which means that they don't need to have air pockets in their bones to make them light for flight. So Kiwi, for the male Kiwi, they're about 2.2 kilos, and the females are slightly larger at 2.8. The reason they're a little bit larger is the size of this egg. So you can see here, just a chicken egg for comparison. We've got, you'll be far more familiar with this egg on my left, and then the Kiwi egg here is typically about 440 grams, so much larger. And what we do when we first bring the eggs to us is we would use a torch to candle the egg, and that tells us what's going on inside the egg. And the keepers here, it takes about 2 years to be a fully trained Kiwi keeper. And by that time, we can interpret the shadows that we see and age the eggs within 1 or 2 days. So if they are viable, we'll have determined that through candling, we will then wash the eggs and move them through into our incubation room. So looking in the incubation room now, we've actually just moved the last egg that we've got of the season through this morning. So this room is actually empty at the moment. Kiwi lay, or brown Kiwi lay, 2 eggs in the first clutch, 2 eggs in the second clutch, and sometimes a third clutch. So we've finished both the first and second clutch for the season, but we may get some more eggs coming back in for that third clutch. So what we do in here is we check the eggs twice a week to make sure that they're progressing as we'd expect. But most of the work is done in those boxes behind us there. So those are our incubators. They keep the eggs at a set temperature of 33.5 degrees Celsius. and assess humidity, and they also turn the eggs for us. And so we need to turn the eggs so that the proteins in the albumin or the egg white get moved around the veins that are on the inside of the egg, and those aren't moving, so we need to move the albumin inside there so that the egg can get the nutrients that it needs throughout development. And of course, those membranes on the inside of the shell are also where you would get gas exchange through the pores of the shell. When the egg gets closer to hatch, it gets to the point where those membranes are no longer getting enough oxygen for that growing embryo inside the egg. And so the bird will actually poke its bill into that air pocket, and then it will start breathing air through its little air sacs or lungs for the first time. So we can actually, as Kiwi keepers, we can hear those eggs vocalize on the bench, which is really something very special. And they'll also be moving. They'll be making that egg wobble on the bench. Now I've got some footage that we can share with you here. This is of Kiwi candling, Kiwi egg candling. So that's that light source being passed through the egg. You can see the veins on the top, the foot kicking here, and you can see the yolk underneath, that yellow yolk. That needs to be internalized in through his navel prior to hatch. Here you can see this one, it's already externally picked. And so for this egg, it's actually moving through to the next room here, which is the hatch room. So you can see on these doors here that this is how the embryo is really quite squashed up in that egg, poking its bill here into what would be the air cell, and it started breathing air through its little lungs for the first time. So this is the last egg that we've got for the season. We may get a couple more, but the last known egg, and this guy is just a couple of days away from hatch. So Kiwi eggs are actually incubated for an incredibly long time. They're incubated for 78 days. Most of the bird species are incubated for about 20 to 25 days. So the incubation time is a long time, and the egg itself is large, and those two things combined mean that when the egg hatches, it is known as a precocious offspring. So that means that this chick is like a miniature adult. It's fully feathered, and everything is instinctive. We don't need to worry about it needing to be taught by the parents, and in actual fact, when this chick hatches, it will only stay in the nest for about 5 or 7 days. So this egg doesn't need any post-hatch care other than keeping it warm for a couple of days while those feathers dry off, and the chick ventures out into the wild. Everything is already known in his head how to probe and forage naturally. So you can see here behind me that we've hatched 81 chicks already this season. We typically hatch about 100 a year. This year's been a little bit quieter, and we think that that's because previous years have been so strong, coupled with a bit of a drought this season has just impacted the number of invertebrates available to those females developing that large egg. And something that's really interesting for those of you that don't know too much about kiwi, it's actually the male kiwi that incubates the egg, because the female, as I mentioned, she can lay up to 6 of these eggs each year, which means it's actually the dad's job to incubate the eggs. So we've had a milestone at our new location. We had not only moved sites, but we also celebrated last year our 2,500th chick, but we're not dwelling on that too much longer because we're already up at 2,600 eggs. And that is purely because of the hard work that the people do out in the field, our team here, and of course our visitors for coming and learning about what we do, and also the revenue that is created from visiting our hatchery goes straight back, 100% back to the Kiwi Hatchery. So what we're going to do is we're going to move through from this egg into the brooder room, because in that room the 2-day-old chick would move into its very own brooder with a heat plate. That heat plate emanates what Dad would be doing for the first week or so. We actually remove it at day 6 because they no longer need any of that extra warmth. And then typically they would feed on their own. So come through and we'll learn about our Kiwi brooder room. So here we have fellow Kiwi keeper, Eve, hand feeding Teatin. Now, you may think, well, I've just said that this bird doesn't need to be taught anything. But that's because in the wild, I mentioned earlier, Kiwi forage in the ground for insects and fallen berries, and insects move. Now you can see that they've got a very long bill, and at the tip of that bill, they've got this extra sixth sense, almost like a spidey sense, and he's using that there to pick up those pieces, doing incredibly well. But in the hatchery, we feed them this special diet, which has been developed by nutritionists at Massey University. And this gives them the very best start to life, because this food has all the micronutrients that the kiwi need when they're growing. In the wild, they'd have a whole ecosystem worth of invertebrates that they'd be able to get all those nutrients. But we use this diet while they're here to make sure that they get that very best start. And because it doesn't move, occasionally our job is to wiggle the food in front of their bill so that they can realise that this is food. You can see here that the chick is actually also quite interested in Eve's fingers, and that's because they've got a pulse. It's being picked up by those sensory pits in the tip of the bill. But other than that, so this chick hatched on the 29th of March. So this chick isn't even 2 weeks old and is already crouching, probing, foraging, so interested, would be out in the wild on its own. But because of that, it does actually make them quite vulnerable. And so this is why we need to keep them safe here at the hatchery. Once they've established onto the food and they have reattained their hatch weight, so we weigh them daily, and we actually see a reduction in weight for that first week or so. And that's as they're utilising that yolk that they internalised prior to hatch. And then we see an increase up above their hatch weight. So the average egg size is about 440. This chick hatched at 404 grams. So this guy came out of quite a big egg and then regained back up post the hatch weight. That's when we will then give the chick a little microchip, same as your cat or dog, and that is for close range identification, so that when this chick goes back out into the wild and then may get refound again, we'll be able to tell whether it was a wild hatch chick, which is actually unlikely because, as I said, those wild hatch chicks unfortunately only have a 5% survival because of those introduced predators, or that it's come through the hatchery. If we want to monitor the chick, we then actually have to put something different on, which is a transmitter onto the leg of that kiwi, and that's how we will go around with an aerial and monitor those chicks in the wild. But we've been doing this work for so long that we know that the survival is really, really good, and we actually want to leave them to it. So we typically don't release them with a transmitter anymore, but if we want to restart the program, we will use specially trained conservation dogs. These dogs, again, it takes about 2 years to train these guys up. They get free flights with Air New Zealand, they get their very own wage, and they actually have a better social media following than I do, Joe. So they're incredibly important dogs in New Zealand, and these specially trained dogs can go out and find the kiwi in the first place because they've got such excellent sense of smell, and then us humans just put that transmitter on so that with a not as good a sense of smell, we can find them. So that's O&E in a nutshell. That is from start to finish, finding the bird, bringing the eggs to us, keeping them safe when they're vulnerable, and releasing them back into the forest where they came from. So any questions?
Yeah, absolutely. Well, that was so cool to be able to see kind of the whole process from coming in to incubating and then hatching and being fed. That's really, really cool. We're going to get some questions coming through in the chat. So if you do have some questions, don't be shy. Use the chat sidebar and send us in some questions. The classes were wondering, I know we can't go into the room, but does the camera zoom a little bit? They're just wondering if they could get a tiny bit of a closer view if the camera zooms.
It doesn't, I'm afraid. Sorry, we've
That's
zoomed
okay.
it in as much as we can. We can perhaps send a photo through for you of a closer image so that you guys can see, because you're right, it is really special. Eve can come to the window now that the feed is finished, so that's I guess an old-fashioned way of zooming, Joe.
Sure.
But she can then see you've got the eyes, the dark eyes, the ear opening behind them, and right at the tip you might be able to see the nostrils at the tip. You can also see those whiskers going into the face, and those strong, powerful legs. So now the food's out of the way, he's trying to get away and go back to bed, so we'll let him go back to bed. Yeah, whilst he thinks he can defend himself, he's only about 300 grams at the moment. And so, yeah, unfortunately, we need to get him 3 times that size, Joe, so that he can then defend himself against the number one predator for Kiwi chicks, which is that stoat.
All right. Well, that was perfect. That worked out really, really well. We got a really good view there. So thank you. Thank you so much. Okay, so questions starting to come in. George would like to know the favorite part. What's your favorite part of your job?
Oh, good question, George. I think my favorite part, I personally love the embryology. We host the National Kiwi Egg Handling course, and I love sharing the knowledge with other Kiwi practitioners and actually even delegates from all around the world. We've had people come from the Smithsonian and San Diego Zoo. So I love sharing what I know about the eggs. You think it's probably the chicks that get most people, but for me it's the eggs. And I think as favorite moments go, it is when I'm on the other side of the glass and I can see visitors watching what I'm doing, and when they see the movement on the bench, suddenly a light bulb goes off and people realize that it's not just an item on a recipe list, there is actually a baby kiwi in that egg, because otherwise as humans, we're not used to eggs, we're mammals, we're used to babies. So yeah, for me it's that seeing the excitement and the passion flick into people's eyes when I see visitors come through.
Yeah, I think it's so cool that they can vocalize inside the egg. I think that would be really cool a really cool experience, and then to have them come out and hatch afterwards. That's really awesome.
That's right. That's right. Great question, George.
All right, our next question. Hunter, can you remind us how old that kiwi is?
So that kiwi hatched on the 29th of March. We're now the 10th today, so it's 13 days, is it? 13 days, my maths is right.
All right, perfect. Well, Pippa's curious about where that kiwi's heading. So how big can they get?
Great question, actually. So I should have mentioned we code all of the eggs. So don't know whether my lovely friend and colleague Nell can get that on the screen. No. So to make sure that we know exactly which egg we're following and the chick, and so that they go back to where they should come from, the first couple of letters of the code is the area, and this one says TK. So I know that's short from Taranaki, which is down on the west coast, southwest coast of the North Island. And then The next couple of letters are short for the male's name, and this male is called Tangaho, which is a name provided by the Taranaki Kiwi Trust that brought in the egg. And then the number afterwards is the number of eggs that that male has brought through the hatchery, so we can see how successful each male has been. So actually the egg that you've just seen in the hatch room is the sibling egg to this chick.
All right, very cool. And so we know that it weighs, the little kiwi weighs about 300 grams. How big will it get in the future? Like when it's fully grown?
So this chick, unlike the graph that I showed you, is still on a little downward trend. So hopefully when they establish onto food, they will reattain their hatch weight, which for this little guy was, oh, I can't see, it's too far around the corner, would have been about 350 grams. And so at that point, they then go to our outside creche area to grow up to that 1 kilogram. And it's all done by weight, not age. And that typically takes about 4 months, something like that. So they're with us for that long. And I should probably also say, Joe, I kind of default and say they're all he. I don't know why. But at the moment, there are no external features for us to tell whether they are male or female. So something else that we do in this room before they go outside is we carefully pluck a couple of feathers and we send that away for DNA feather testing. And that will tell us whether they are male or female.
All right, very cool. Well, you just answered Levi's question. He was wondering how fast they grow. So about 4 months and they'll reach that 1 kilogram. Margot wants to know, how big is the kiwi's beak going to get?
Great question. All of these questions are so good. So before we had DNA feather testing, we actually tried to determine the sex of the bird by the bill length. And so the bill, if it is over 100 millimeters, they used to say that was a female. But of course, if it was shorter than that, we wouldn't know if it was a juvenile female or a male. So typically they can get that long, and it is definitely proportionate to their body, and the females are typically slightly larger, if you remember, than the males being 2.2, females 2.8 on average.
All right. Harrison was wondering if the eggs are pretty standard in size, or do they kind of vary? Can they be bigger or smaller?
So many great questions there. They can actually vary quite a lot. So even the shape of the egg can vary. So sometimes it's a lot pointier at the non-air cell end. So that air cell end is typically the blunt end. They can be quite torpedo shaped, and those are typically younger females that haven't laid many eggs. And similar as you might see eggs categorized by size 6, 7, and 8 in the supermarket, your chicken eggs, the kiwi eggs are exactly the same, that the older the female laying the egg, she will typically lay larger eggs. And so the largest egg that we've had has been over 500 grams, and the smallest egg has been around 220, I think it was. So there's a lot of variation, and you can also have variation in shell color as well. So the shell is actually 95% calcium carbonate. So that final 5% can be down to the diet of the female. And so, yeah, sometimes, most of the time they're white, but sometimes they can have a bluey green hue as well.
All right. Last question for you, Emma. After the different clutches are laid, is there a downtime where there's no eggs in the hatchery? And if so, what's going on at the hatchery?
Oh, so there's a bit of a downtime for the kiwi out in the wild. So typically their calendar year starts about April, and they will call to each other, because for the brown kiwi, whilst they stay in mostly monogamous pairs, they're not always found together in the same nest, but they do they've got a territory that they defend together, and they will call to each other. And the most calls start in April. They then sort of settle into starting to lay eggs in June. We start seeing the first eggs coming to the hatchery August, September. By this time, as you said, we're now sort of waiting for that third clutch to come along. And so after the third clutch, there is potentially a little bit where kiwi themselves get a break. But for us at the hatchery, unfortunately, not so much of a break. They're certainly peaks in season. So when you guys had brought DARWIN200 to New Zealand back in October, that's definitely a peak, and that's why we were focused on chick rearing then. But yeah, there are quieter times, but there's usually the chicks from the previous season are still on site growing up to that kilo when we start getting the eggs from the next clutch in.
All right. Well, Emma, thank you so much. It's been a while since we've been able to hang out and do one of these events together. So always fun to catch up and visit the hatchery, especially in the new location. It looks incredible. Obviously, a huge shout out to you and the whole team for the incredible conservation work you do. And a shout out to the team members who join you today, our friend behind the camera, as well as behind the glass. So great to have them with us today to make this happen.
Yeah, thank you, Joe. Really appreciate the opportunity. And of course, time-wise, I do hope everybody has a wonderful Easter. It's very fitting to work with eggs and chicks. So yeah, give us a thought over Easter and think about how you might be able to help us save kiwi.
All right, amazing, Emma. Thank you so much. We're going to say goodbye to New Zealand for now, but I hope you have a great rest of the week. Thanks, Emma.
And you. Thanks. Bye.
All right, very cool. So great to connect with Emma and some of the team at the hatchery. Really great tour, really great signal, and really nice to have that nice close-up view of that young kiwi, barely reaching 2 weeks old. That's pretty awesome. All right, well, we have a few things to do today. We still have that video coming up that Josh and I recorded together from the ship in the harbor in South Africa. We have an experiment to look at from 2 weeks ago, our growing cress seed experiment, and then we have the answer to this week's curiosity. There won't be a new curiosity and there won't be a new experiment because I know classes are going out for a few weeks, so we'll pause. Once we get going again, we'll start again with some more curiosities and some more experiments of the week. Let me go ahead and share my screen. So if you remember our experiment for the cress seeds, we were trying to grow them in 3 different environments. So with lots of light and some fresh water, with fresh water, but then covering them so the light wasn't coming through. And then our final one was lots of light, but using salty water. So I'm going to share my screen here. It should come up in just a moment. And let's take a look at that results together.
Welcome back, everyone. Let's have a look at the results of our seed germination experiment. As you might remember, we put some cress seeds on a towel in fresh water and exposed it to sunlight. A towel in fresh water covered up with this plate in darkness. And over here, we put some cress seeds on a paper towel with salt water. Well, let's see how the different experiments compare in terms of the germination rates. Well, we can clearly see that the cress seeds with the fresh water exposed to sunlight have germinated very, very well. There's hundreds of little cress plants growing up here. This one here in the middle, exposed to dark conditions, well, these have also grown very well, but you can see the cress seeds aren't as green and their long, thin stems have been snaking around in different directions. Last but not least, our saltwater experiment, well, actually these ones have barely germinated at all. A few of them have tried to germinate, but none of the seeds really have taken off, and yeah, the ones that have tried to grow don't look very healthy at all. Well, what does this experiment show? Well, clearly, the seeds that have germinated the best are the seeds that have been on the freshwater and the paper towel exposed to sunlight. So clearly, cress needs both fresh water and sunlight in order to grow healthily and the little seedlings grow strongly. The seeds that have been put in these dark conditions have germinated, but they're not looking that healthy. So this reveals that cress seeds absolutely can germinate just with fresh water without sunlight, but the plants, in order to grow healthily, need sunlight in order to photosynthesize. Interestingly, if you can imagine when the plate was on the bowl, the seal isn't perfect and a tiny little bit of light would have probably shone in through the sides. And that's reflected in the growth pattern of these little seedlings. You can see them all growing out sideways, towards that tiny little bit of light that maybe came through. So it shows two things. Firstly, that cress seeds can germinate even if there's no light or very, very little light. They just need the fresh water in order to be stimulated to do so. But when the seeds germinate, the seedlings search out for light and do their best to grow towards any light source, as we can see here. The reality is, if we kept these little seedlings in the dark, they would eventually starve and die because they need to photosynthesize in order to grow healthily. And that shows the difference between these ones here in the sunlight and these ones here in the darkness. And our last experiment over here has the most dramatic results. Very few of these seeds here that were put in the salt water have germinated, and the few that have mostly died or are looking very unhealthy and I think won't survive. So this reveals that clearly salty conditions are not good for cress plants or cress seeds to germinate. Remember that every single different species of plant around the world has different requirements for its seeds to germinate and for its seedlings to grow healthily. Some plants actually like salty conditions. These plants are called halophytes, and if you used the seed of one of those, you'd probably end up with the exact opposite results, where the seeds wouldn't probably grow in fresh water, but would grow very well in the salty conditions. You can repeat this experiment and test different types of seeds. See if you get different results with different species of plants. Send in your results to classroom@darwin200.com. It'd be really, really interesting to see if you got the same results as I did here with cress, or if you find out different tolerances with different types of plants. Thank you for taking part.
All right, there we go. That was the result of our experiment. As you can see, the light and the fresh water, it grew beautifully. Surprisingly, maybe surprisingly for some of the students, the one completely covered still grew really well. In fact, it grew longer, but that's because those cress seedlings were desperately searching for light. So they were growing longer, they were spreading out, they were trying to find a source of light. Maybe there was a tiny little bit of light coming through the edges, and so that's where they were kind of pushing. But they would have been a little bit paler. They wouldn't be as green as the one that was growing nice with lots of light. And then the saltwater conditions, well, no good for growing those cress seeds. They didn't really germinate at all, and there really wasn't any growth. So we do have a $50 Amazon gift card for a classroom. I'll reveal that classroom by sharing what they sent to me with some of their results. So, whoops, there we go. Bear with me while my screen shares. You should see it now. So there we go. How will they grow? So they, our class here in South Australia, they did a great job listing the materials. You can see they've got Stu there in the background. So they were probably watching the experiment, making sure they were doing all the steps and not missing any. You can see that they got their Petri dishes ready. They got the paper towels nice and damp, sprinkled the cress seeds over it, and then it was time to put some in the light, to put some in the dark, and then to put some salt water in those last ones. So there they go. They set them up in their new classroom location to see what would happen. So they had a little slowdown. I guess there was air conditioning in the room, and that kind of dried out their seeds. But as all good scientists do, they persisted. And over the period of time, they were able to get some action. So they're able to get some growth in those Petri dishes. So what did they find? Very similar to Stu. So the one where it just had water and light, it had green shoots, it was growing. The ones that were in the dark, some of them still germinated, but they were pale green color. And then finally, the cress seeds with the salty water, they didn't tolerate the salt. They didn't grow at all. So a big shout out to our students who joined us and sent in this great experiment results for us. Very, very cool. Okay, I'm going to come back from that screen share. There we go. And now we're going to spend a little time with Josh. So I connected with Josh on the ship. V&A Waterfront is where the ship is docked right now. And let's see what Josh had to say. And maybe, just maybe, there was a little bit of wildlife in the harbor as well. So let's take a look at this video with Josh together. Here we go. Let's see if we can grab Josh in here. Let's see how Josh is doing today. Hey, Josh. Josh, can you grab the mute? We still got you on mute. No worries. No, that did not. Oh, now you're okay. No, Josh, we don't have your sound. Oh, wait. Yeah, we got it now.
Just froze for a minute. Yeah, great.
No, we got you now. Josh, good to see you.
Yep, good to see you too. How's things?
Yeah, good. Good. We just had Amy from Marine Dynamics. She's one of their marine biologists. I don't know. Do you know what your project is next week yet?
Next week I'm doing the Cape Vultures.
Okay.
Out in the mountains, about 3 hours away from Cape Town, and I'm not sure what I'm doing on the second week yet. I've not been told.
All right.
But it sounds like whoever ends up with the penguins and the sharks is going to have a great time.
Yeah, it's a beautiful spot, but getting out there with the vultures, that sounds pretty cool. So I'm looking forward to seeing a little bit of footage.
Yeah. I'm looking forward to it.
Okay, so you made it. We left you maybe 2 days before arrival. Anything of note before dropping anchor in the harbor?
We had an amazing day of arriving. We woke up and the weather was a bit rubbish. It was really cloudy. You couldn't really see much land, so we weren't expecting much of the arrival. And then we had the wildlife started showing up. So we had 2 southern right whales that were playing with each other right next to the ship.
Wow.
And we had enough time to watch them for a bit, run downstairs, get the drone up in the air and film them. So they were really hanging around for a while. And then we had some dolphins join the boat, and then there's the fur seals that we see all over the place. And yeah, it's been quite a windy few days, but the weather today is beautiful. It is hot today.
Yeah.
So yeah, maybe a bit too hot for me, but I think some people are quite enjoying it.
All right. Well, Cape Town is a pretty iconic harbor. Can you see anything from there? I see cranes behind you. Is your view pretty restricted, or can we see any of the mountains?
No, I mean, we've got, I'm not very good at navigating the laptop camera, but if I go like, there you go. There's Table Mountain in the background there.
Table Mountain, yeah.
And then interestingly, we've got this, it does look very much like an industrial port here.
Yeah.
But this ship here, this is the
Agulhas 2? Yeah.
Yep, and this is the ship that they used to rediscover the wreck of the Endurance, Ernest Shackleton's ship. And we didn't know that was going to be here until we showed up and we watched a documentary about it on the way over. So it's a really nice coincidence that it's there.
That is so cool. We did Exploring by the Seat of Your Pants, my organization. We did all the live events from the ship while they were looking for it.
Oh, wow. That's really cool. I didn't know that.
Yeah. So with any luck, it'll still be there. Yeah, that's really cool.
Yeah. And awesome. other than that, I mean, there's a lot going on wildlife-wise in this harbour. I think there must be a lot of fish because we've got loads of sea lions jumping around today. Yeah, you can see a pile of sunbathing sea lions here.
Oh, yeah, I can see their heads. Yeah, one swimming by pretty close.
Not a sea lion, sorry, a fur seal.
Yeah, having a little play.
Yeah, and we've got the gulls and the terns are diving into the water a lot, so there's obviously a lot of fish coming to the surface, maybe because of the heat.
All right, very cool. Well, have you been able to explore a little bit of the city at all? I know you have a few days.
Yes, we actually got to do, because we arrived early, as to give the guests something to do, the Oosterschelde arranged a tour of Cape Town, so we got to go down to a penguin colony, and down to the Cape of Good Hope and up to the top of Table Mountain and stuff. So we really got around and we really saw as much as you can see in a day. It was really nice.
Yeah, that sounds great. Josh, what I want to do now, I have that sizzle reel of the arrival. So I'm going to play it. And if you want to maybe just narrate a little bit while it's playing. You definitely see an overcast day.
Yeah. So this flag was morning. That time in the morning. Yeah, and there were a few of these boys all along the way, and they were all covered in those fur seals. And I'm not sure what species of dolphin that is, but we had a few dolphins coming and going, and then these are the 2 right whales that we saw. As you can see, they're really close to the shore. Yeah,
from what Amy was saying, it's a pretty cool encounter because I think it's more more around May when they really start to come back in bigger numbers.
Oh wow! Well, yeah, it was awesome to see them. Really, that's the closest I've seen them. To see them sort of interacting the way they were interacting for as long as they were was really nice.
Yeah, so it's hard to tell from the video because you had the drone up, but how close to the ship would you say they were?
When we sailed past them initially, they were probably, I don't know, 150, 200 meters away from the ship. But by the time I got the drone up, we were already sort of well past them.
Yeah.
But like I said, they stuck around on the surface, flapping their fins around long enough for me to run downstairs and get the drone in the air. So, yeah, it was just really nice to see. Really nice encounter.
Yeah, you're a pro with the drone now, so I imagine the launching and landing from the moving ship are just second nature now.
Yeah. And let's hope it stays that way.
Yeah, absolutely. Very cool. Great. Well, so you've got a few days. Then the Darwin leaders are going to come in. You're heading about, you said 3 hours away for the Cape Vultures. That sounds pretty awesome.
Yes. Yeah. It looks like a really interesting project, and it sounds like we're working with a few of the local people around as well to give us better access and better encounters with the birds. So yeah, I'm really looking forward to that.
Yeah. Do you know offhand? Have you had a chance to talk to Rolf? Do you know how many leaders arrive next week?
I do not, I'm afraid. No, I've got no idea. I can tell you at least 3 because I know there's me, Tom, and another camera operator coming, but maybe more. I've got no clue. I would imagine more.
3 for sure, because I know there's going to be the 2 heading down Hans Byway. You're going to be out with the Cape Vultures, so yeah, at least 3, but we'll see. You never know what Rolf has up his sleeve.
I know, yeah. But no, it'll be interesting to meet some of the Darwin leaders we're getting in as well, because we're getting, as far as I'm aware, we're getting a lot of African Darwin leaders coming to South Africa because it's so hard visa-wise to get them to other continents.
Yeah.
We've almost been saving up all of the African applications for when we get to South Africa, so it'll be really nice to work with a different demographic of people.
Yeah, no question. All right, Josh. Well, I won't keep you too long. I know it's hot on deck. I know they're doing a lot of work. I'm sure you might want to jump off and explore, or maybe you're editing the voyage video.
Yeah, I wish I could jump off and explore, but I'll be on my laptop for the rest of the day.
Yeah. All right. Fair enough. Well, Josh, we always love getting to steal you for a little bit.
Yeah, thanks. It was great catching up.
Yeah. Well, have fun with the Cape Vultures. I will see you probably sometime in a couple weeks.
Yeah, great. Looking forward to it.
Awesome. Thanks, Josh.
Thanks. All the best. Bye-bye.
Yeah. See ya. Okay, there we go. A little catch up live in the V&A waterfront. Shortly, Josh will be heading out about 3 hours north, and he'll be out in the field with a young Darwin leader. He'll be the camera operator. They'll be out with a conservation team, and they'll be looking at the Cape Vulture. So hopefully we get some really cool pictures, some footage from in the field, and we'll be able to share that at some stage as well. Well, it looks like we're going to wrap up a little bit early tonight, which we don't do very often, but that's because we don't have a new experiment or new curiosity to share. However, we do still have to take a look at our curiosity from last week. So if you'll give me a moment here, we will get that loaded up front and center. And let's take a look at the answer to last week's curiosity.
Last week, I posed the question of what group of organisms barnacles belong to. Did you have any ideas? Could you work out which group these strange animals belong to? For hundreds of years, naturalists argued over this very question. For a long time, many people thought they actually belonged to the mollusk family. That's the family that includes shellfish, squid, octopus, and other organisms like that. There are many, many species of mussels that grow anchored to the rocks, so it's not difficult to see why barnacles were thought to belong to mollusks, especially because many of them produce small, hard shells that superficially look similar. But if you look at barnacles really closely when they're underwater, for example in a rock pool when the tides come in, you might see something very strange happen. At the opening here at the top, a strange limb-like appendage unfolds and actually filters the water for particles, which is what the barnacle feeds on. No mollusks have an appendage like that. So Darwin, during his study of all of the barnacles known at that time, when he wrote a monograph of of all the barnacle species, worked out that they actually belong to crustaceans. That's the group of organisms that includes crabs, lobsters and shrimps. Darwin noticed that over millions and millions and millions of years, a shrimp-like organism had actually evolved into the barnacles that we see today. One important bit of evidence that added weight to Darwin's argument was the fact that barnacles have free-swimming larvae just like other groups of crustaceans and unlike mollusks. So we now know that barnacles are actually a type of crustaceans. It's amazing to think how a shrimp-like organism could evolve into these hard structures that we see today, anchored to rocks right the way around the world. Did you guess correctly? Let's find out now who won this week's competition. Thank you for taking part.
All right, let's get that nice and full screen. Okay, so there we go, the barnacle. Really, really cool creature. Darwin wrote a whole book on them. If you look at Darwin, he was a pretty prolific writer, whether it was writing about earthworms, evolution, barnacles, the list goes on and on and on. He had a lot of thoughts about a lot of different things, carnivorous plants. So, no shortage of ideas and thoughts, and that 5-year journey that we're recreating in 2 years. He saw some amazing places, some amazing wildlife, plants. Yeah, what an experience for a young naturalist, and it really set him up for the rest of his career. So Miss Fitzpatrick's crew, some 3rd grade students in Cairns in Queensland, sent in a correct answer this week. So we have a gift card for your class, and I will get Sophie to send one of those your way. And then I thought we might end off a little bit early, but I was reminded in the chat that we did do an experiment last week, but we had a really short time frame for it. We did it in 1 week, but it was a really easy experiment. So classrooms were able to get that done. So I'm going to squeeze in that results video right now. I should have it here handy. So bear with me for a second, and we will take a look at that results video together. Here we go. We'll talk about the experiment, and then we'll talk a little bit about why it happened. So this week's experiment is another, and I'll just turn the volume off because I'm going to be the one doing the talking. So here we go. We have an experiment where I filled a plastic bag with water. I used a Ziploc bag. And then I had some pencil crayons with me. You could have used pencils, maybe in your classroom or at home. So there we go. Plastic bag, nice and full and bulging with water. And then I had those pencil crayons with me. Then I took those pencil crayons and I put them, there they are. And one by one, I put them through the plastic bag. So I used my secondary camera here so we get a nice good view of it. And I wanted you to see there was no tricks going on here. These pencils were actually going through the bag. All right, there goes the first one. As you can see, there is no water pouring all over my desk, which is really good because I have a lot of electronic stuff on my desk. So it's a good thing that the science worked out in this experiment. All right, so as we're watching a few more of those pencil crayons go through the bag, let's talk about why this is happening. So there's a few things at play here. We got to talk a little bit about plastic bags. We got to talk about polymers to understand that this wasn't magic, this was science. So we'll start off by talking about polymers. So polymers are long chains of molecules, and they make up many things that we use every single day, specifically plastic items. So every day, I mean, if you look around your classroom, you look around your home, you're going to find a lot of plastic items. And so polymers, like you find in your Ziploc bags, your plastic baggies, also in grocery store bags, plastic water bottles, soda bottles, food packaging, they're all made of polymers. And polymers are great because they're flexible, they're lightweight, they're leak-proof, and they're cheap to produce. So that's a reason why polymers make up plastics. They have many advantages. So they're kind of like a long chain of beads. So when I took the pencil crayons and I put them through the bag, it pushed those polymers apart. So it went right through the chains of beads. Because they're so flexible, they were immediately able to push back towards the pencil and make a temporary seal around that pencil crayon. So that prevented the water from leaking out those holes and from spilling all over my desk. And then when I took the pencils out later over the sink, all the water poured out because the holes were way too big after the pencil crayons were removed to redo that seal. So polymers, they're long chains, kind of like beads. They are flexible, lightweight, leak-proof, and low cost, which is why we see them in so many plastic items. And that is why when I put the pencils through the bag, they were able to quickly reseal with that flexibility around the pencils, and I did not make a mess on my desk. Now, of course, there's lots of other issues we can think about with plastics and plastic bags, and that is we use far too much single-use plastics. Plastics were developed and meant to be used in items that were supposed to last a really, really long time. But because they were so cheap to produce, we started using them in things that we use once and throw away. But luckily, in different places around the world, we're seeing a move away from plastic bags and straws and single-use bottles, which is great because we know that those usually end up in our ocean in a lot of cases and then can take thousands and thousands, tens of thousands of years to actually degrade into smaller and smaller pieces. They do take a long time to break down. All right, there we go. That is our last experiment before the break. We had several classrooms give that experiment a try. I thought we kind of might because it was a nice and easy experiment. So Esperance in West Australia, Mr. Gleason's crew, we have a gift card for you this week, which is great. I also have a submission that I will share because I know that they're joining us right now. They already scored a gift card for their cress seeds, but I know our crew in South Australia is definitely here with us live right now. So I'm going to share their submission as well because I know they always like to see their stuff on the screen. So here we go. Let me share here. So it looks like it was a nice demonstration for the crew. They went outside and did the experiment. If we look at those pictures, you can see those pencil crayons going right through at different angles. It looks pretty cool. And it was great to hear that there were some other kind of science and thoughts that came out of it, like looking at the way it looked like the pencil crayons were bending because of refraction through the water. So this crew is definitely thinking about what was going on. They talked about polymers. They talked about flexibility and sealing as well. So that's great. Awesome. So 3 classrooms taking part in that experiment. Okay, for real this time, we are going to wrap up our event for today. A huge shout out to all of the classrooms who join us on camera, who join us live and then who join the recordings afterwards. It's great to have you with us. And I hope you have a great break. And then I'm going to have so much South African content to share with you. You won't believe it. We're going to have lots of live stuff, lots of videos and things like that. So I can't wait to get out there, start collecting all that material, and then share that with everybody upon my return and when the break ends. The last thing we have to do today is a shout out to our sponsors, our supporters, our partners, without whom we wouldn't be in South Africa right now, sailing around the world. Our Darwin leaders wouldn't be arriving shortly. We wouldn't have the world's most exciting classroom and the research on board the vessel. The Oosterschelde would not be happening. So that's where we're going to leave it for today. Hope everybody's a great Easter, a great break, and we are going to sign off for now. Thanks, everyone.